Yeast genome. Yeast genome consists of 16 chromosomes. In addition to 16 chromosomes, there is mitochondrial genome as well as a plasmid of 2 micron. So the genome of the yeast consists of 16 chromosomes, which is present in the nucleus region. In addition to chromosomes, there is a mitochondrial genome as well as a small plasmid, which is called as 2 micron plasmid. The haploid yeast genome consists of approximately 12.1 MB of DNA. Yeast genome was completely sequenced by 1996. If we see the characteristics of the yeast genome, it is small and compact. There are small intergenic DNA sequences, means the DNA sequence between two genes, which is called as intergenic DNA sequences. There are few transposable elements which are present in the genome of the yeast. Few introns have been present in some of the genes of the yeast, and there is limited RNA interference that has been observed in the genome of the yeast. Yeast genome is predicted to contain approximately 6,200 genes. In addition to these genes, there are 274 transfer RNA genes. There are 287 introns. Small percentage of yeast gene have introns. The intergenic space between different genes can be up to 200 base pair and 1000 base pair. So, in some of the genes, the space between two genes, it can be 200 base pair, and in some of the genes, the space between two genes, it can be up to 1000 base pair. If we do the comparison of different characteristics of the yeast genome, we see that the yeast genome consists of chromosomes, plasmid, and mitochondria. So the relative amount of chromosomes is 85%, plasmid is 5%, and mitochondria is 10% of the total genome of the yeast, including all these three chromosomes, plasmid, and mitochondria. If we see the number of copies, as the yeast contains 16 chromosomes, and in case of plasmids, there can be 60 up to 100 copies of the plasmid, and in case of mitochondria, the number of copies can be in between 8 to 130, and on average, the number of copies of the mitochondria can be 50. If we do the comparison of the size of the genome between chromosomes, plasmid, and mitochondria, in case of chromosomes, the size of the entire genome of the chromosomes is approximately 12,100 kilobase pair, or in other words, we can say that 12.1 MB of DNA. In case of plasmid, it can be 6.3 kb, and in case of mitochondria, it can be between 70 to 76 kb of the DNA. So this is the comparison between the genome of the chromosome, genome of the plasmid, and genome of the mitochondria, because all these three are present in the cell of the yeast. The largest known regulatory sequences which are present with a specific gene sequence, they are up to 2800 base pair, and the name of the gene is MUC1. So these are one of the largest regulatory sequences which are present with one of the gene of the yeast genome. Yeast gene has been named consisting of 3 letters and up to 3 numbers. These are the examples G, P, D. So these are the 3 alphabets or 3 letters, and there can be up to 3 numbers. So GPD1 is the name of one of the gene which is present on the genome. The second example is HSP12. So here we can see that there are 3 alphabets and there are 2 numbers. Usually these are meaningful. They have the meaning concerning related to the function of those genes or the genes which are related to different pathways of the genome of yeast. Wild type genes which are present on the genome of the yeast, they are usually written as italics and in capitals. Here we can see that TPS1 is written in capital as well as in italic. So these genes, they are wild type. While in case of recessive mutant genes, they are also written in italics, but they are in small alphabets. Like the example is small TPS1. So this is a recessive mutant gene of the yeast genome. The 3 letters provide the information about the function, mutant, phenotype or the process related to that gene. So here we can see that CDC is the name of the gene which is related to cell division cycle. C is for cell, D for division, C for cycle. And ADE is for ADE9, that is ADE9 biosynthesis. In addition to nuclear genome or the genome of 16 chromosomes, there is a small mitochondrial DNA which is present in the yeast cell. So this is the size of approximately 75 kb, although the size of the mitochondrial DNA can be between 70 to 75 kb. Here this is the typical example of the mitochondrial DNA. We can see that on the mitochondrial DNA of the yeast genome, there are genes for the respiratory complex, there are genes for the ribosomal protein gene, there can be introns. Here we can see that there are introns, the genes for the ribosomal RNA genes, then transfer RNA genes and some other genes. So these are, let's say, these are the transfer RNA genes. Here we can see that these are the transfer RNA genes. Respiratory complex gene, these are respiratory CO2 and CO3, these are respiratory complex genes. Here we can see VAR1, that is a ribosomal protein gene. And there are also introns, ribosomal RNA genes. They are also present in the mitochondrial genome of the yeast. In addition to mitochondrial DNA, in the yeast, there is a small plasmid DNA. That is 2-micron circle. This is the name of that plasmid, 2-micron circle, and the size of the plasmid is 6.3 kb. There can be 50 up to 100 copies per haploid genome of the yeast. So this plasmid can be between 50 to 100 copies. There are genes for ARS, FLP, and 3 genes which encode a protein required for the regulation of FLP. Here we can see that this is ARS. Here is the gene for FLP and REP1 and REP2. These are genes and the D gene. So these genes, they are required for the regulation of FLP. Small set of direct repeats which are called as STB, they are also present here. STB is also present in the plasmid DNA. These small direct repeats are usually used during the process of mitosis and meiosis and the cell division of the yeast cell. So if we conclude that what is yeast genome? Yeast genome consists of 16 chromosomes. In addition to 16 chromosomes, there is a mitochondrial genome as well as a small plasmid which is also present in the cell of the yeast.